Welcome to this tutorial video. So in today's topic, we'll look at what we call glycogenolysis. So before you watch this video, I hope you have watched my video on glycogenesis. If you haven't, just make sure that you watch that video first, because this video makes a lot of sense if you have watched my video on glycogenesis. So, glycogenolysis. Now, this is a topic that involves the breakdown of glycogen back to glucose. You can see from the word itself, you've got glycogen, then you've got lysis. So what does it mean? It's a biological process that involves the breakdown of the stored glycogen back to glucose. Now, it is important for you to understand why this particular process is occurring. So, when your blood glucose levels are low, right? When you are low on blood glucose levels or low on energy, what your body does is that it will start utilizing the stored glycogen, that glucose that you had stored into glycogen in excess. But because you are low on energy now, it will start breaking down the stored glycogen in the muscles, in the liver, back to glucose, so that your body can utilize it for the needed functions. Remember that your brain, okay, it highly depends on what? On glucose as its fuel to perform those different cognitive functions. Talking of red blood cells, they need, glucose. Talking of your muscles, they need glucose. And other, organs that need this particular glucose. That is why when you are in the fed state, remember in glycogenesis we said that excess glucose will be stored to glycogen for what? Energy storage purposes. But now, since the energy levels, or rather the blood glucose levels are low, we'll start now breaking down this glycogen back to glucose. And this is what we refer to as glycogenolysis. Right. So, looking at the key terms, we have to define this officially, and we're saying that glycogenolysis is a process by which stored glycogen in the liver and in the muscles is broken down into what we call glucose 1-phosphate and eventually glucose 6-phosphate, which can produce energy. It is a process which involves the breakdown of stored glycogen. Where? In the liver and in the muscles. Remember, when we were studying glycogen in glycogenesis, we said it can be stored in the liver and it can be stored where? In the muscles. So, those are the many organs. So now we start breaking it down back to glucose. Don't worry about the glucose 1-phosphate, glucose 6-phosphate. I will explain how these particular molecules are formed so that we can produce energy from them, or what we call glucose. Now, if you don't want to use that particular definition, you can simply say glycogenolysis is a process of breaking down glycogen to produce glucose. And when the glucose is produced, okay, you must understand that that glucose now is going to undergo glycolysis and to be able to provide energy for the body. Even better if there is, aerobic respiration, because you know that from that glucose that we're going to produce, it will now form pyruvate. Pyruvate, in the presence of oxygen, it will form what we call acetyl-CoA, which can initiate the Krebs cycle, and from the Krebs cycle, you're going to produce your electron carriers. We said NADH and FADH2. And those now are going to proceed to what we call electron transport chain, and they'll provide your body with the needed ATP energy. So that's what we are looking at. But I need you to also know that glycogenolysis is a separate pathway. It is not the reverse of what? Glycogenesis. I know you may try to pair the terms since I'm talking about glycogenolysis, which is breaking down, and you may start pairing it with glycogenesis. No. Glycogenolysis is a separate pathway and not the reverse of glycogenesis. Alright. Now, let's look at the key enzymes. Let's look at the key enzymes of this glycogenolysis process. Now, on your left here, you've got our key enzymes. The first key enzymes, actually we've got three key enzymes. The first key enzymes, and it is the rate limiting enzyme that I'm mentioning. I'll explain why. We've got what we call glycogen phosphorylase enzyme. Glycogen phosphorylase enzyme. This particular enzyme breaks what we call alpha 1,4 glycosidic bonds. So you remember that in my glycogenesis tutorial video, I told you to say, when you are forming the straight chain unit of the glycogen, you use what we call alpha 1,4 glycosidic bond. And in that particular session, we said the enzyme that was forming, those particular bonds was what we call glycogen synthase. But now, since we want to break down this glycogen back to glucose, what do? We must break these alpha 1,4 glycosidic bonds using what? Glycogen phosphorylase enzyme. Apart from that, we've got what we call glucan transferase. You can see here, we've got what we call glucan transferase. So this enzyme doesn't do really much, but it's very, very important. It transfers what we call a trisaccharide unit of glucose from one branch to the other. I'll explain how this enzyme works. It transfers a trisaccharide unit of glucose from one branch to another branch. Apart from that, you remember to say in my glycogenesis class, I told you to say a glycogen molecule has got branches. And for us to be able to release a free glucose molecule, we must break down those particular branches. So what is the enzyme, sir? The enzyme is what we call debranching enzyme. Remember in glycogenesis, when we were forming those branches, we were using what we call a branching enzyme. But since this time around, we want to break down this glycogen. So we use what we call debranching enzyme. But what type of bonds does this debranching enzyme break? It is what we call alpha 1,6 glycosidic bond. And when that bond is broken, we are going to release one glucose molecule from each particular branch. So, I repeat, the key enzymes we've got what? Glycogen phosphorylase enzyme. It breaks 1,4 glycosidic bonds. We've got glucan transferase. It transfers a trisaccharide unit of glucose from one branch to another. I'll explain this. Debranching enzyme. It breaks alpha 1,6 glycosidic bonds. Because I know that especially if you are doing health-related courses, you must understand that a glycogen molecule has got what we call alpha 1,4 glycosidic bond and alpha 1,6 glycosidic bonds. No wonder we are using these particular enzymes to be able to break it down. You must also know the key regulators of this particular process. You must know the key regulators of this particular process. And among those regulators, we've got what we call glucagon. So, you and I know very well to say glucagon is a hormone that is released by the pancreas. What does it do? You've always been told that glucagon promotes the breakdown of glycogen back to glucose. What does it tell you? It simply tells you to say glucagon will promote glycogenolysis. Apart from that, I also need you to know, I'll explain how this particular amino acid is formed, the CAMP. It simply means cyclic adenosine monophosphate. Now, this molecule actually regulates both glycogenosis and glycogenolysis. But you've seen that I've put a negative sign on glycogenesis, meaning it inhibits glycogenesis and it promotes glycogenolysis. Apart from that, you must also understand that epinephrine, or what we call adrenaline. I know you've always been taught from your background, grade 12, biology, to say adrenaline, or what we call epinephrine, prepares the body for fight and flight. When somebody is frightened, when you are scared, or when your body is under what we call stressful condition, you release what we call adrenaline. Why? Because your body is trying to prepare you for strenuous activities. Because you may want to run. For example, you're walking on the road, then you see a snake. You want to run because you are frightened. And now what your body will do is, since it knows that after that you are in a stressful condition, you may need energy to be able to run. You may need energy to be able maybe to kill the snake or do something. It's going to release this epinephrine so that it will promote the breakdown of what? The stored glycogen back to glucose, of which you know very well to say from that glucose you will be able to obtain ATP. Even better if it undergoes, when you form pyruvate and it undergoes aerobic respiration. So epinephrine, or rather what we call adrenaline, it's going to promote what we call glycogenolysis, as well as your growth hormone. So growth hormone, for you to be able to grow, okay, you need nutrients. You need a lot of, physiological function that you need energy and ATP. So growth hormone is going to promote the breakdown of what? glycogen back to glucose. So that glucose now, if it undergoes glycolysis, it will be able to provide the body with the needed ATP. So we've looked at the key enzymes. We've also looked at the key regulators. Actually, I'm going to emphasize more on the key regulators as well as we conclude this particular session. Now, let's have this recap. So, what you are seeing on your right here is basically a glycogen molecule. Remember, this is how a glycogen molecule that we had formed in glycogenolysis looks like. It has got a glycogen in there, okay, this protein, that is linked to straight chain units of what? Of glucose. And I need you to remember that these straight units of glucose are made up of what we call alpha 1,4 glycosidic bonds. And I've already told you the enzyme that breaks these particular bonds in glycogenolysis. We've said it's what? Glycogen phosphorylase enzyme. Now, you say, sir, this particular glycogen, it also has branches. Yes, because glycogen is always stored in it has got the branching. It's formed it's always stored in a branched form. It has got branches. No wonder we said these branches, okay, are made up of what we call alpha 1,6 glycosidic bonds. You've got a branch here, you've got a branch here, you've got another branch here. But we already have a solution. Why? Because I've already told you to say these branches will be broken down by what we call the branching enzyme. Don't worry, I'll explain to you how the glucan transferase works. I just needed you to remember that this is how a glycogen which has been stored in the liver or in the muscles looks like. And for us to be able to break it down, we are going to use those three enzymes that I've mentioned: glycogen phosphorylase enzyme, glucan transferase, and the branching enzyme. So in the next slide, we will see now how we'll be able to manipulate this particular molecule and break it down so that we can release the needed glucose into the bloodstream, okay, and so that it can do the needed, physiological functions. So let us look at this particular slide. So you know very well to say glycogenolysis takes place both in the muscles and in the liver mainly. So I'm going to use the liver to explain this particular process, but I also emphasize the things that you have to know about the muscles. Now, you must understand that for you to be able to break down this glycogen molecule, it's the same slide that it's the same slide here. I've just added, the specific enzyme. So for you to be able to break down this, stored glycogen, you start from the branches. You will not start from the stem like here. No, you start from the branches because this is more like a tree. So you're going to start from the branches to break it down. So let's see now how the glycogen phosphorylase is going to work, how glucan transferase is going to work, how the branching enzyme is going to work, and how we will be able to produce that free glucose that the body needs to raise what? The blood glucose levels. Because remember, at this particular point, the reason why you are breaking down this stored glycogen is to raise the blood glucose levels because they are low. Alright, so let us start. Allow me to zoom in just a bit so that you can see properly. So, we'll start with glycogen phosphorylase, then we'll go to glucan transferase, then the branching enzyme at the end. So let's see how now this glycogen phosphorylase, my boy here, okay, it's glycogen phosphorylase enzyme. How is it going to help in breaking down this stored glycogen? And remember, it's taking place where? In the cytoplasm. Because when we had stored it in glycogenesis, we had stored it where? In the cytoplasm. So we'll go back and start breaking down from the very cytoplasm of the cell. Now, this glycogen phosphorylase enzyme, how it's going to break, I've indicated for you to say, in this straight unit of, glucose chain, this straight unit, this has got what we call alpha 1,4 glycosidic bond. So I've just reminded you by indicating here. So how are we going to break these particular bonds? How are we going to break them? So the alpha 1,4 glycosidic bonds, as we've already agreed, they are broken down by glycogen phosphorylase enzyme. So how does it do that? So this glycogen phosphorylase enzyme, it will use what we call phosphate molecules, okay, or phosphate groups, okay. It has got these inorganic phosphates. So what it will do, you can see here, there's even a 5 for you, just to remind you to say, it will get about 5 phosphate groups from its pocket, the glycogen phosphorylase enzyme, what is it going to do with them? It will get 5 phosphate groups. It will sprinkle them. It will sprinkle them on this straight chain unit of glucose that is linked by alpha 1,4 glycosidic bonds. Don't worry about these lines on your left. We're just trying to remind you to say at this particular molecule, there's a 6th carbon of this glucose unit. But the bonds that you form in the straight chain unit are what? Alpha 1,4 glycosidic bonds. So what will glycogen phosphorylase do? It will get about 5 phosphate groups. It will sprinkle them on this chain. When it sprinkles them now, that phosphate group is able to break the alpha 1,4 glycosidic bonds. It's going to break them. So let's say it breaks about 5, okay? But you must understand to say that phosphate group that was sprinkled there will just not disappear. It will attach itself to what? To the carbon atom number 1 of this glucose molecule. So if you sprinkle the phosphate groups here, those phosphate groups, all of them, all the 5 that you've sprinkled, all of them will attach to each glucose, molecule at carbon atom number what? Number 1. So you remember, when you attach a phosphate group at C1 carbon of a glucose molecule, you call it what? Glucose 1 phosphate. Glucose 1 phosphate, because you've attached a phosphate group at that particular carbon of this glucose molecule. So if you sprinkle on the 5 different bonds, okay, on the 5 different alpha 1,4 glycosidic bonds, you release 5 glucose 1 phosphate molecules, because each phosphate group is going to attach itself to the first carbon atom of this glucose molecule. So what glycogen phosphorylase is doing is breaking the alpha 1,4 glycosidic bonds, and it's releasing the glucose 1 phosphate. Glucose 1 phosphate. Why? Because those phosphate molecules sprinkling on this chain, they are attaching themselves to the first carbon of a glucose unit, and we are forming what we call glucose 1 phosphate. We will see how that glucose 1 phosphate is going to be used later. Now, apart from that, you must also understand that this glycogen phosphorylase, it's limited. No wonder we call it the rate limiting enzyme. Its activity is limited. How? I want you to focus here. So we've got a branch here, right? We've got a branch, and this branch has got what we call alpha 1,6 glycosidic bonds. So let's count from down. Let's count our glucose molecules. Let's count 1, 2, 3, 4. So this glycogen phosphorylase cannot go beyond the 4th glucose molecule to break this alpha 1,4 glycosidic bond. So if it reaches the 4th one, it has to stop here because it's limited. Okay? It has to go to another branch and sprinkle. So if let's say it comes on this branch, it can't go beyond 1, 2, 3, 4. It can break any bond here, but it won't go beyond the 4th one. So after it's done breaking these, alpha 1,4 glycosidic bonds, if it reaches the 4th glucose, it has to go to another branch and do the same thing. So we've just remained with what? The 4 of them here on each branch. That's where now my enzyme here that you can see, my ninja here, glucan transferase, it will be like now it's my 10. So what will glucan transferase do? This glucan transferase, what it will do is that, remember I said it transfers a trisaccharide. Tri means 3. So you've remained with 4 here because glycogen phosphorylase was not able to go beyond the 4 glucose unit. You've remained with 4. But since we're saying glucan transferase transfers a trisaccharide, it will just get 3 of them. So from 4, you remove 3, you just remain with 1. So glucan transferase is going to transfer these 3. 1, 2, 3. It will end here and just leave this, 1 glucose molecule here. So when glucan transferase, cuts the bond here, okay? Cuts the bond here, it has got what we call alpha 1,4 glycosidase effect. It will be able to cut here. So it is going to get this trisaccharide unit and attach them on this particular branch, just to elongate it, just to give this glycogen phosphorylase a lot of, chance to be able to break those bonds, because if there are 4 glucose units remaining, it can't go beyond that. So glucan transferase will just be trying by all means to elongate the chain to give glycogen phosphorylase a lot of opportunity to be able to break these bonds. Now, after glucan transferase, transferred this trisaccharide unit onto this chain, you remain with 1 glucose, this 1 glucose molecule here. But you say again, sir, yes, we've remained with 1 glucose molecule, but it's still attached to the branch. And this branch has got what we call alpha 1,6 glycosidic bonds. Now, what do we do with the branch? Then the debranching enzyme will be like, I'm here, I'm going to do this particular job. The debranching enzyme is going to break what we call alpha 1,6 glycosidic bond. But remember that on this end, all right, glucan transferase has already moved or transferred these three onto the other chain. So meaning on this end there's nothing. And then the branching enzyme has also broken this bond. So meaning at this particular point now, you're going to have a free glucose molecule, which will now be released, okay? And do the needful. If it's needed in the brain, it's going to be transferred to the brain through the bloodstream. So that's how you're going to release a free glucose molecule. What have we said? Glycogen phosphorylase sprinkles the phosphate groups. I mean, sprinkles the phosphate groups onto this alpha 1,4 glycosidic, bond of this particular chain. Then it's going to release glucose 6,1 phosphate. Why? Because the phosphate group is being attached itself on C1. Then since it can't go beyond the fourth glucose molecule, okay, in this chain, glucan transferase is going to come transfer a trisaccharide unit onto another branch. Then you just remain with one glucose molecule. This one, since it's still attached to what? To a branch, the branching enzyme is going to it's going to break the alpha 1,6 glycosidic bond, and you release your free glucose molecule. So that's how glycogenolysis takes place. So from there, glycogen phosphorylase is going to move to another branch, do the same. Glucan transferase is going to follow, do the same thing. The branching enzyme is going to follow, and it will be releasing those particular free glucose molecules. However, you must know that, look, from each branch, regardless, even if this was long, you'll be able to release only one free glucose molecule. But let's say if your blood glucose are very low, okay? Let's say they're about, 3.9 millimoles per liter of blood. That particular patient that you may be dealing with is in what we call hypoglycemia. So you need a lot of glucose. But there's a problem. Why? Because from each branch, you just release the last one here, the last glucose molecule. You come to this branch, you just release one. You come to this branch, just release one. So we must play around that. No wonder I've put even a ratio here to say every 5 glucose 1 phosphate. Remember, 5 glucose 1 phosphate is being made from glycogen phosphorylase. You only release one glucose molecule. This ratio is not good for the body because you need a lot of glucose. Now, how are we going to play around? Because we must manipulate now this glucose 1 phosphate back to glucose somehow. Now, in the liver, this is possible. You can convert glucose 1 phosphate into glucose in the liver, but in the muscles, this is not possible. So we will understand, or we will know how the muscles are going to play around that particular part. So let's assume that our glycogen phosphorylase keeps on doing the same thing, It's releasing what we call glucose 1 phosphate. Let's now zoom in here. So let's assume we've got our glucose, 1 phosphate already present in the cytoplasm. So how do we form now glucose? So glucose 1 phosphate, remember in my glycogenesis class, I told you to say this step when you're converting in glycogenesis, when you're converting glucose 6 phosphate to glucose 1 phosphate, it was reversible. So meaning you can go back even in here. So glucose 1 phosphate will be converted back to glucose 6 phosphate. What enzyme are we going to use? The very enzyme that we used in glycogenesis, phosphoglucomutase enzyme. So you convert a lot. Imagine this glycogen phosphorylase, it keeps on sprinkling these phosphate groups, breaking the branches. So you've got a lot of glucose 1 phosphate. But what you want is glucose. That's why now this glucose 1 phosphate will be converted to glucose 6 phosphate using phosphoglucomutase enzyme. Lucky enough, in the liver, you can convert glucose 6 phosphate back to glucose. So regardless of the glucose 1 phosphate that are coming in the liver, you can form glucose. Why? Because glucose 6 phosphate will be converted to glucose using what we call glucose 6 phosphatase. So what this particular enzyme does, it's going to remove that phosphate group at carbon atom number 6 of this glucose 6 phosphate, and you're going to release your free glucose. That's why the liver is known for providing our bodies with what? Free glucose if the need arises. Because it can convert glucose 6-phosphate back to glucose. Then by now, you would have played around that and you provide your body with the needed glucose that it wants, and you'll be able to raise, the blood glucose level back to normal. So that's how you do that. Now, I said glycogenolysis also takes place in the muscles. So in the muscles, everything that I've mentioned here is the same. However, your muscles cannot form glucose. Why? Because they lack what we call glucose 6-phosphatase enzyme. That is even why in my gluconeogenesis session, I told you to say muscles do not undergo gluconeogenesis. Why? Because they don't have the glucose 6-phosphatase enzyme. So your muscles are going to do this part. They can convert glucose 1-phosphate to glucose 6-phosphate, but they can't convert glucose 6-phosphate to glucose. Why? Because they lack glucose 6-phosphatase enzyme. So what will your muscles now do? Because they also need energy. Remember, the actin myosin mechanism in muscle contraction need what? ATP. So how will they do that? Since they have managed to form glucose 6-phosphate, I'll remind you, in glycolysis we said the first step is to convert glucose to glucose 6-phosphate. We have our glucose 6-phosphate here. So what your muscles will now do, they'll redirect this glucose 6-phosphate into the glycolysis pathway, into the glycolysis pathway, so that it can now undergo glycolysis and produce ATP. So your muscles, I repeat, can't form glucose because they lack glucose 6-phosphatase enzyme. But they can form glucose 6-phosphate, which they will just redirect to what we call glycolysis so that they can produce ATP from that. That's how we break down this particular glycogen and provide free glucose to the body. All right. Now, let's take note here. There are differences of which I know I've already mentioned them, but there's a difference between liver glycogenolysis and muscle glycogenolysis. Okay. Now, don't worry about this part. I've already explained, because you've got your glucose 6. You've got your glucose 1-phosphate. It's at carbon atom number 1 here, and then you've got your phosphoglucomutase. So this one, this step is just this part is just trying to remind you to say this is present in both, because both muscles and the liver can convert glucose 1-phosphate to glucose 6-phosphate. You can see that the phosphate group is at carbon atom number 1 here, but this phosphate group is at C6, carbon atom number 6. So this part is there, but the differences are that in the liver you've got glucose 6-phosphatase, which removes the phosphate group and releases free glucose into the blood for other organs and tissues. That is in the liver. I've already talked about this. In the muscles, there is no glucose phosphatase enzyme. So what your muscles do, they will use glucose 6-phosphate into the glycolysis pathway and make energy for running, or maybe for activities, or whatsoever reason that these muscles will need this particular glucose for. So that's that part. Now, let's talk about the regulations. I know I've already talked about them, but let me just emphasize a bit. Now, glycogenesis regulation. Remember I said glycogenesis is supported by what we call insulin. Insulin promotes glycogenesis. That is why in diabetic patients, when they've got a lot of increased, when they've got increased blood glucose levels, we give them insulin. Why? Because insulin is going to promote glycogenesis. It will be storing now that excess glucose in the blood back to what, or into what we call glycogen. Now, you must understand, how does insulin do that? How does it promote glycogenesis? This is how. So you've got your liver cell here. You've got your liver cell here. So when you give a patient insulin, even your body, for those people that are not diabetic, they have got insulin, which regulates their blood glucose level. So this mechanism is the same, whether it's in a diabetic patient or in somebody who has got normal insulin levels. The mechanism is the same. So how does it promote glycogenesis? So insulin, if released, it will move through the bloodstream. It will go towards this liver cell. Now, on this liver cell, we've got what we call tyrosine kinase receptor. Tyrosine kinase receptor. So this receptor is going to respond to this insulin. Imagine this is insulin. It will respond to this insulin. When tyrosine kinase receptors respond to this insulin, it will be converted to what we call protein phosphatase enzyme. Protein phosphatase enzyme. Now let's hold on a bit here. You must understand that. Remember, I said the rate limiting enzyme of glycogenesis is what? Glycogen synthase. The rate limiting enzyme of glycogenolysis is what? Glycogen phosphorylase. So what happens is that this glycogen synthase is active without phosphate, is active without phosphate groups around it. It doesn't want phosphate groups at all. So if it has got phosphate group or a phosphate molecule on it, it will be deactivated. But you know that glycogen synthase is the one that is used to form glycogen, right? But if it's deactivated or it's not active because it has got phosphate molecule on it, then that will be a problem. So what insulin is going to do now is that since it will make tyrosine kinase respond to it, this tyrosine kinase receptor will form what we call protein phosphatase. Then this protein phosphatase now will start removing those phosphates. So it will start dysphosphorating what we call glycogen synthase. It will remove all the phosphate groups around it or the phosphate molecule around it. Why? Because this phosphate group, they make it not to be active. You can see it is active without phosphate. That's how now insulin makes that protein phosphatase. And then these now are going to remove those phosphates. So that's how come glycogen synthase is going to be active, and it will now be converting the excess glucose into what? Glycogen. I repeat, if you give insulin, okay, or if insulin is released in the bloodstream, it will react with what we call tyrosine kinase receptor. Okay, that conformational change will make this tyrosine kinase receptor to form protein phosphatase, which removes phosphates. Remember, the phosphates, they deactivate glycogen synthase. It's always active without phosphates. Then they are going to remove these phosphates and make it active so that it can promote glycogenesis. Now let's talk about degradation of glycogenolysis. Now, in this case, we use glucagon. Remember, glucagon promotes glycogenolysis, or the breakdown of what? Glycogen. So how does it do that? Now, I told you to say the rate limiting enzyme of glycogenolysis is glycogen phosphorylase. And let me remind you, glycogen phosphorylase, what was it doing? It was using phosphate groups to sprinkle on the long chain of that glucose unit, which had 1,4 alpha glycosidic bonds. So, meaning glycogen phosphorylase, for it to be active, it needs phosphate group. It's the opposite of what glycogen synthase needs. This one, glycogen phosphorylase, needs phosphate groups so that it can sprinkle them. So how does glucagon now promote this process? What glucagon is going to do is that when released into the bloodstream, it will go to the liver cell, of course. Then this receptor now is not tyrosine receptor. This one is what we call G protein coupled receptor. So the G protein coupled receptor will respond to glucagon. Since this one needs phosphates, what will happen is that this G protein coupled receptor will form what we call adenyl cyclase. This adenyl cyclase will convert ATP into cyclic adenosine monophosphate. Cyclic adenosine monophosphate. This cyclic adenosine monophosphate is going to form what we call kinase A. This kinase A enzyme now is going to start adding phosphates. No wonder I told you to say cyclic adenosine monophosphate promotes glycogenolysis. Why? It adds phosphate groups on what? Glycogen phosphorylase enzyme. Because glycogen phosphorylase enzyme need phosphate groups for it to break those alpha 1,4 glycosidic bonds. But if this, kinase enzyme add phosphate in glycogen synthase, it will deactivate it because this one doesn't need phosphates. So that's why cyclic adenosine monophosphate promotes glycogenolysis by adding phosphates onto the glycogen phosphorylase enzyme. So that's how this process of glycogenolysis regulation works. So this is the end. I hope you've learned something on this particular process of what we call glycogenolysis. Don't forget to like the video and also subscribe to this particular channel. Thank you so much.